Assalamualaikum and hi, I'm your Hamiza Binti Abu Bakar, S76691 from Group 8. So now I will talk about the effect of storage. For introduction, we studied the effect of storage time on the bacterial count of various food samples. Our group was assigned a fresh fish sample at 0 hour storage time. Storage time is one of the important factors that influence the microbiological quality of food. As food is stored for a longer period, microorganisms, especially bacteria, can multiply and increase the risk of spoilage. Fresh fish is considered a highly perishable food because it contains a high amount of moisture and nutrients that support bacterial growth. As a result, fish can spoil rapidly if not handled or stored properly. To determine the bacterial population in the sample, the total plate count or TPC method was used. This method allows us to estimate the number of viable bacteria present in the food sample by counting the colonies formed on agar plate after incubation. In this experiment, the fresh fish sample at 0 hours served as the baseline sample, deparesting the entire bacterial load before any extended storage period. The result obtained can help us understand how storage time affects bacterial growth and food quality. This experiment is important because it helps us evaluate the microbiological quality of food sample and understand how storage time affects bacterial growth.
Assalamualaikum, my name is Nushika binti Nurdin, metric number S76813. I will be explaining the methodology. Our group used fish as a sample with several materials and equipment with each stomacher bag, 2 bottles of 225 ml of sterile suhlin, 5 tubes containing 9 ml sterile suhlin for serial dilution, 3 PCA, pipettes, and incubator with set at 35 Celsius. First, add 225 ml of sterile suhlin into the stomacher bag containing the fish. Second, homogenize the mixture by placing the bag in the stomacher machine for 2 minutes. This mixture is now your 10 -1 dilution. 3. Prepare a serial of dilution tubes containing 9 ml of sterile suhlin. 4. Pipette 1 ml from the 10 -1 stomacher bag into a 9 ml saline tube to create the 10 -2 dilution, and then mix well. 5. Repeat the serial dilution step by step, transferring 1 ml into 9 ml of saline until you reach the required dilution factor for your group. 6. Pipette 1.1 ml of the dilute sample into a PCA or an air plate. 7. Spread the 110 ml in column evenly across the agar surface using a sterile glass spreader, which is hockey stick. 8. Invert the plates and incubate them at 70 Celsius for 24 until 48 hours. 14. After incubation, make a preliminary estimate of the number of colonies on all 3 plates. 15. Count and record the number of colonies for your group, choosing only the plates that fit the standard counting criteria, which is 30 until 300 colonies. 16. Calculate the total plate count and report the final value in colony forming unit. Show your sample calculation for the report. 17. Share and combine your group data with the rest of the class. 18. Discuss the effect of storage time on the bacterial growth of fresh fish based on your group and class result.
For discussion, I will explain the effect of storage time on the microbial count in fish. For group 3, it uses fish sample left at room temperature for 4 hours, and it had numerous mixed population of bacterial colonies formed because the fish is being left at room temperature for 4 hours, and the room temperature is known as danger zone, and because the room temperature falls in the range of 5 degrees Celsius to 60 degrees Celsius, and 4 hours of exposure to room temperature has caused the bacterial growth because the room temperature is suitable for supporting bacterial growth, allowing maximum rate of enzymatic activity and biochemical metabolism. Therefore, bacteria reproduce and multiply effectively. Lag phase is shorter than exponential phase. Then the group 4, which uses fish sample left at room temperature for 12 hours, it had the greatest growth of bacterial colonies compared to the bacterial colonies formed in other groups such as group 3, 8, and 9, because the fish use because this fish is being left at the room temperature for very long hours compared to the other groups. Therefore, this fish had the most longer time of exposure to room temperature compared to other fishes in the other groups. Therefore, this condition has allowed the bacteria in this fish to have the highest amount of time to grow and multiply rapidly compared to the bacteria in other fishes, because longest exposure of fish to room temperature has provided the most suitable environment for rapid exponential bacterial growth, and bacteria was able to reproduce and multiply actively, and these bacteria had the shortest generation time. Then the group 8, which uses the fish sample left at room temperature for 0 hours, it showed the lowest bacterial growth compared to other groups because the fish was not exposed to the room temperature at all, and therefore it was not placed in the danger zone. Since it was not in the danger zone, it lacks the sufficient time and temperature exposure, which is an important fatum factor, and this has caused the bacterial growth to be limited. However, some microbial growth still occurs because the fish is a highly perishable food that contains high level of nutrients, moisture, and water activity, and this factor has provided a favorable environment for microbes, allowing the bacterial growth to still occur even the time and temperature conditions are not optimal. Then the fish sample from group 9 showed a higher number of bacterial colonies than the fish sample from the group 8 and group 3 because it was left at room temperature for 8 hours, and this prolonged exposure has caused the fish to be in a long exposure of danger zone, allowing the bacteria to be provided with sufficient time to multiply rapidly with favorable condition, allowing metabolism to be efficient. Now I'll explain the relation between the sample storage time and the dilution factor selection. First, the fish sample left at room temperature for 0 hours was expected to have the lowest microbial load. Therefore, the lowest dilution factor has been used to make sure sufficient colonies to be detected and counted, and using higher dilution factors will lead to error result because it will cause a few or no colonies to present due to initially low bacterial population. Then the fish sample left at room temperature uh, was expected to have a higher microbial load than the fish sample left at 0 hours because of the fish being exposed to room temperature, creating favorable condition. Therefore, a moderately higher dilution factor is used to obtain countable colonies, as using lower dilution factor is unsuitable because it will result in overcrowded plate, making the colony enumeration to be difficult. The fish sample left at room temperature for 12 hours was expected to have the highest microbial load due to the most prolonged exposure to danger zone. Therefore, it involves the use of the highest dilution factor to reduce the microbial concentration to obtain a countable and well-separated colonies. The use of lower dilution is not suitable because it will lead to overcrowded plates, causing too numerous to count result and making the enumeration difficult.
So me, Firdaus, S76211 will be continue for result part. For 0 hour storage, we started with fresh fish, testing low dilution from 10 minus 1 to 10 minus 3. The result, the 10 minus 1 plate show 190 colonies, giving us a baseline for normal bacteria on fresh field. The 10 negative 2 and 10 negative 3 plates were TNTC. This suggests a minor plating error but confirm bacteria is actively present. At 4 hours, higher dilution, which is 10 minus 2 to 10 minus 4, were used to account for expected bacteria grow. Every single plate was TNTC, even when dilute 10,000 times, the bacteria grew so fast that the colonies completely merged together. By 8 hours, the fish was spoiling, so the sample would dilute up to 10 million times, which is 10 minus 5 to 10 minus 7. For the result, the 10 minus 6 plate gave a clear count of 260 colonies, proving a massive spike in bacteria since hour 0. The other two plates were completely overgrown, which is TNTC, confirming extreme bacteria levels. For the last one, which is 12 hours storage. At 12 hours, extreme dilution, which is 10 minus 6 to 10 minus 8, were required because the fish was heavily spoiled. For the result, despite being diluted for 100 million times, all plates were TNTC. This shows massive, completely unchecked bacterial multiplication by the end of the test. So me, Firdaus, S76211, will be continue for conclusion part. Our group analyzed the 0 hour raw fish sample, representing the free hash condition. The sample show the lowest microbial count compared to the 4-hour, 8 hours and 12 hours sample. This indicate that microbial growth was minimal at the beginning of storage. As storage time increased, microbial count also increased. Therefore, shorter storage time helps maintain the freshness, quality and safety of raw fish.